Chapter 5, Skin. In today's lecture, we'll talk about growing organs in a laboratory setting. These will use a patient's own stem cells, but will require taking the fascia from a cadaver. We'll talk about skin color and how and why we have different skin tones. We'll talk about melanoma, one of the more deadly forms of skin cancer. We'll talk about vitamin D. It's often said that this comes from the sun, but in reality it's just the energy that comes from the sun. So we need to understand where vitamin D truly comes from. We'll talk about what does cellulite look like. We'll talk about cleavage lines. This will be very important for anybody going into surgical technology. And we'll also talk about stretch marks, which are a type of scar tissue, and why some of them might appear darker and others lighter. In today's lecture, we'll start off with membranes and fascia, and then we'll move on to the skin, covering first the epidermis and then the dermis. The last bit for today will be how wounds heal in the skin and why we might have scars left over. Before we get to the skin, we've got a couple of things that are more complicated than tissues, but not complicated enough to be considered organs. The first are membranes. Membranes consist of two tissues, an epithelium plus a little bit of connective tissue. There are four types of membranes: the mucous, the serous, the cutaneous, and synovial membranes. I'll cover the basics right now. The rest of today's lecture is about the cutaneous membrane, and synovial membranes will be covered in more detail in Chapter 9. Many organs contain two membranes. A membrane is an epithelium plus a connective tissue, and we generally do not classify these as organs. They're parts of organs. For instance, the gastrointestinal tract contains a serous membrane on its superficial surface, and the deepest surface is a mucous membrane. Mucous membranes, oddly enough, produce mucus. They consist of an epithelium and connective tissue. We find them in the digestive, respiratory, urinary, and reproductive tracts. Mucus can trap pathogens, aiding in their removal from the body. It can also lubricate things moving through tubes, such as feces. In case you're wondering, there are two spellings to the word mucus. One is technically an adjective, the other is a noun. Many anatomy books do not recognize this distinction, so you will see both spellings in different places, often used incorrectly. Next up are the serous membranes. These are very important in our ventral body cavity, like we discussed in Chapter 1. They include a simple squamous epithelium plus a little bit of connective tissue. The epithelium is called a mesothelium, and of course when this one is mutated to form a tumor, we call it mesothelioma. The most common cause of mesothelioma, at least in the past, was exposure to asbestos. Now that we understand this connection, we have reduced the risk of this, at least here in the United States. There are a number of other countries where asbestos mining is still legal, and we purchase asbestos from them. We find these types of membranes in the ventral body cavities, including the pleura, the peritoneum, and the pericardium. The cutaneous membrane is a topic of today's lecture, coming up in a minute. And lastly, the synovial membranes are very important to the joints that move. We'll talk about these in greater detail in Chapter 9. Fascia are also considered to be more complicated than a simple tissue, but not complicated enough to be considered their own organ. These consist of just connective tissue. But if you look over a broad area and notice an overall organization of the collagen fibers, We call this organization fascia. The fascia are a 3D web of extracellular matrix found between organs. They help to anchor one organ to the next. We can classify fascia based on their location. There are superficial, deep, and subserous fascia. I do not have any of these on models in the classroom, so you will not be tested on these names in the lab section. When fascia become damaged, organs in one part of the body can protrude into another part of the body. This is called a hernia. The most common type of hernia is an inguinal hernia, where the intestines protrude into the scrotum. This can block blood flow to that portion of the intestines, which leads to cell death, which leads to inflammation and pain. Fascia are just the connective tissue portion of organs. Organs can be removed from cadavers and stripped of all of their cells, leaving behind these collagen fibers and fibronectin. This framework can be used to grow stem cells to replace organs. So far, this has been successful in bladder replacements and a few isolated other organs, but will probably become a lot more common in the near future. So those are the membranes and fascia, two things that are not considered to be organs, but are nevertheless important. The rest of the lecture will focus on the skin. We'll cover its basic functions, and then its two major layers, the epidermis and the dermis. And then we'll talk about some physiology, skin color, vitamin D production, and the formation of scars. To start off, the skin is the largest organ in the body. It's made of two major portions, one part that we call the cutaneous membrane, and then the rest of the accessory structures. The cutaneous membrane consists of the epidermis and the dermis. Below that is the hypodermis, which is not considered part of the skin. It is loose connective tissue underneath the skin that helps anchor it to underlying structures, such as muscle tissue. Frequently, this is subcutaneous fat. Accessory structures include hair, nails, and sweat glands. All three of these are epithelial in nature. Let's start with the epidermis. It's a very thin layer of stratified squamous epithelium. It's avascular, just like most of our epithelia. So it cannot be too thick because it relies on diffusion of nutrients from the underlying dermis. We can classify skin based off of whether it contains thick or thin epidermis. The old-fashioned name is thin skin and thick skin, although the more technically correct name would be thin epidermis and thick epidermis. In thin epidermis, there are only 4 visible layers of keratinocytes, whereas with the thick epidermis, found on the palms and soles, we can find an extra layer to the epidermis. Now, the epidermis, either way, is still a stratified squamous epithelium. But in thick skin, it's a little bit thicker than in thin skin. As you can see here, this is a little bit thicker than this. Luckily, these are both under the same magnification, so we can compare these two. Now, 100 years ago or so, somebody looked at the stratified squamous epithelium, not knowing that it was a stratified squamous epithelium, and said there appeared to be more than one layer to this multiple layered epidermis, and they gave those layers names. At the base, we've got the stratum basale, then spinosum, granulosum, potentially a lucidum, and then eventually at the most apical surface, the stratum corneum. Personally, I'm less interested in the names of these layers than I am in their functions. So let's focus on the physiology. In the stratum basale, this is where we find the stem cells, the epithelial cells that are capable of undergoing mitosis and producing new epithelial cells. The stratum basale, like the rest of the epidermis, is avascular. So nutrients must diffuse through the rest of the epidermis. For this reason, the epidermis cannot be very thick. So this is the location where new keratinocytes are produced. From here, new keratinocytes get pushed more apical, and as they do, they start to fill up with keratin, and as that happens, they start to die. And after about a month, those cells will be dead and close to the surface of the body. Two conditions that can affect the stratum basale include psoriasis, which is a condition where there is an excessive amount of mitosis here. This can be treated with drugs that block mitosis. Next up is keratosis pilarsis, where the amount of mitosis is fine, but the amount of keratin that is produced is excessive. This can lead to reddish patches showing up on the backs of the arms. Next up in our list of layers is the spiny layer, or the stratum spinosum. The keratinocytes here are beginning to dehydrate, and their cytoskeleton is beginning to poke through. Hence, they appear spiny. Next up is the grainy layer. The cells here are beginning to produce keratin, and they are starting to die as they fill up with this protein. In thick skin, next would be the stratum lucidum, a layer that cannot be seen on the microscope. Hence, we're not going to be too concerned with it, other than its name. Lastly is the stratum corneum. The cells here are completely dead. They are really nothing more than a little bit of phospholipid bilayer and the protein keratin. This is what constitutes the waterproof layer of our skin. And if you're in the state of Oregon, you understand very well the difference between waterproof and water resistant. You may remember from the previous lecture that stratified squamous epithelia could either be keratinized or non-keratinized. This means that the cells at the surface are either dead because they are nothing but keratin, or they are still living. The non-keratinized epithelia line some of my internal surfaces, whereas the keratinized epithelia line my external surfaces. Where the epidermis and the dermis meet is not a smooth surface, it is bumpy. We call these bumps dermal papillae. It is bumpy to increase surface area, and this increases the strength of the connection between the epidermis and the dermis. For the same reason, you might rough up a piece of wood with sandpaper before you glue it to another piece of wood. This would increase the amount of glue between the surface of the two pieces of wood. When you layer on top of these dermal papillae a layer of epithelium, and then another layer, and then another layer, ultimately these microscopic dermal papillae become bumps that are visible at the surface of the fingertips and toes, called the epidermal ridges. These are responsible for our fingerprints and toe prints. A blister can occur when the two layers of tissue become separated from one another, and fluid builds up between the epidermis and the dermis. Because the epidermis is reliant on the dermis for nutrients, the epidermis above this blister would very quickly begin to die. Here are a couple of pictures showing the epidermal ridges as they are visible from the surface of the skin. As you may notice, the sweat pores are highly organized. The distance between one sweat pore and the next is not random, but very strictly controlled by what we called in the previous chapter planar polarity. The epidermis contains some cells that are not epithelial. These include the Langerhans cells, which are part of the immune system. I will cover these in more detail next quarter. Similarly, the sensory cells that can be found within the skin, I will cover in greater detail in BI 232. So that covers the basic layers of the skin, as well as what we find in the epidermis. The main function of keratinocytes is to produce the protein keratin. As these cells get older, they produce more and more keratin until eventually the entire cell is nothing but keratin. There's no nucleus, no endoplasmic reticulum, no anything else. These keratin fibers are cross-linked to one another, and this ultimately produces a very tough and water-resistant barrier at the surface of this epithelium. Because it is water-resistant, this prevents water from evaporating from our body. Nevertheless, it is only water-resistant, not waterproof. We do lose some water from our body every day. Most of that is lost in our urine. Next up, a lot of water vapor is lost from our lungs, but a small amount is lost from sweat and even insensible perspiration. This is water loss across the stratified squamous epithelium, as opposed to out of a sweat duct. The epithelium in our fingers can become wrinkly when immersed in water. Our textbook may try to suggest to you that this is due to osmosis, depending on which book that you have, but that is wrong. In fact, the fingers shrink when immersed in water. That is because this induces vasoconstriction. We know this because people whose nerves have been cut to the fingertips do not experience this wrinkling when their fingers are immersed in water. The color of our skin is dictated by three major pigments. Those include carotene, which is an orangish pigment found in vegetables, which after we eat it winds up in our skin. Secondly is melanin, which is made in our skin. This is a brownish or a blackish pigment, depending on the type of melanin. And the third is hemoglobin, which is a maroonish color found underneath the skin. There are some other pigments that contribute to our skin color, but the three from the previous page are the three major pigments, and those are the ones that I would like you, or should I say, I will demand that you remember. Before we move on, let's consider the difference between carotene, which is a pigment found in our skin, but comes from vegetables such as carrots, versus the molecule keratin, which is made in our skin and provides our skin with water resistance, but provides no color. These two sound similar, but have very different functions, so please do not get these two confused. And by please, I mean you're going to lose points if you do. So our skin color comes from these major pigments. Of the three, only melanin is actually made in the skin. It is made by cells called melanocytes, found in the stratum basale. Everybody has roughly the same number of melanocytes. People who have darker skin, their melanocytes make more melanin and a darker type of melanin. Those of us who have freckles or moles, those are regions of the skin that produce more melanin, not necessarily melanocytes, just more active melanocytes. Here are a couple pictures of skin. You will notice that on the left, we see somebody with darker skin. They are producing a significantly larger amount of melanin. All of those darker staining cells are dark because of the melanin. Nobody had to add any sort of purple or pinkish stain to make those appear darker. However, not all of those are melanocytes, as we will see in upcoming slides. That is just extra melanin. The melanocytes are the cells that are responsible for making melanin, but they can give that melanin to the keratinocytes. Down in the stratum basale, the melanocytes make melanin, package it up into tiny little granules, and these granules are distributed to the keratinocytes. As these keratinocytes get older, they move closer to the surface of our skin, but they retain this melanin. For that reason, all of our skin cells, the keratinocytes and the melanocytes, should have some melanin to help protect them. The major function of melanin is to absorb UV light. It is not to give our skin color. There are any number of molecules that could give our skin a particular color. The reason that we have situated upon melanin is that it absorbs UV light. One reason why the body would want to absorb UV light is that when it hits DNA, it can cause mutations, and mutations can lead to cancer. We do not want to overproduce melanin because UV light, despite being a class 1 carcinogen, meaning something that we know causes cancer in humans, is that we also require a certain amount of UV light to activate vitamin D, which is required for calcium absorption and good bone health. Now that we've learned a little bit about the epidermis, we can discuss skin cancer. The focus of this class is always basic physiology. So when we discuss cancer, I would like you to refer to what we've already learned. Basal cell carcinoma and squamous cell carcinoma are both very common forms of skin cancer, but they're rarely ever lethal. And that's because the cells, even when they become mutated, typically remain anchored to other cells by the anchoring junctions, or desmosomes. Therefore, these skin cancers are easy to remove by a simple surgery. Melanoma, on the other hand, is not so simple and is quite often lethal. This is a cancer of the melanocytes. These are not actually epidermal cells. They come from the nervous system and migrate into the skin. Because these cells are migratory, when melanocytes become mutated and start to form a tumor, that tumor very frequently metastasizes or moves to other parts of the body, making this type of cancer much more difficult to treat and much more likely to be lethal. The rates for skin cancer in the US are different depending on how much melanin you have. For those who self-identify as white, the risk of melanoma is significantly higher than those who identify as black or Hispanic. One common way of trying to identify melanoma early is by looking at the ABCDEs of moles. Moles are local accumulation of melanin, not necessarily an increased number of melanocytes. Nevertheless, a mole should be fairly regular in its appearance. If it looks asymmetrical, if its borders are uneven, if it has more than one color, or if it's too large, or if it changes over time, that might be a sign of melanoma. Usually, however, it's not. And most of the time that people do get melanoma, it does not arise from a weird-shaped mole. So you may ask, why do we do it? Well, it's the best test that we have. It's very non-invasive and cheap. So it may not always detect melanoma accurately. At least it's an inexpensive and easy test to do. Melanin was one of our 3 major skin pigments. Its production is under the control of a hormone called MSH, or melanocyte stimulating hormone. It turns out, even though we have different skin colors, we have the same number of melanocytes, and we all produce roughly the same amount of MSH. So what exactly contributes to our skin color is complicated. We call it epigenetic. For instance, darker-skinned parents tend to have kids with darker skin, and lighter-skinned parents tend to have children with lighter skin, although there's no guarantee. And it's not a gene that we pass down, like we pass down genes for eye color and hair color. So while it's true that those of you with higher levels of melanin are at a reduced risk of getting skin cancer, that's not why melanin production evolved. Even melanoma would rarely kill people before they had a chance to reproduce, passing on their genes and epigenetic markers. A much better hypothesis is that we evolved melanin to absorb UV light, just like we said, in order to protect folic acid levels in the bloodstream. It turns out that for lighter-skinned people, just 1 hour of sunlight can cut folic acid levels in half. If you have had a child, you may have taken a folic acid supplement during your pregnancy, and that was to prevent very serious birth defects like spina bifida, which can be caused by a folic acid deficiency. So our skin color is both a question of nature and nurture, meaning we do inherit our skin color from our parents, although it's not genes but epigenetic markers. But our skin color also responds to the environment, namely, those of us who get exposed to more UV light, our skin tends to get darker. So this process of tanning also involves melanocytes and the hormone MSH. And it's a homeostatic regulatory mechanism. The more UV light that we are exposed to, the darker our skin becomes to absorb more UV light. When UV light hits our skin, it can cause mutations to the DNA in the living keratinocytes. When this happens, these keratinocytes can release the hormone MSH, and it travels just a short distance. Diffusion does not travel very quickly, so only nearby melanocytes will get this hormone, and they start producing more melanin and depositing this melanin into the new keratinocytes in the stratum basale, and thus the skin gets darker. If you want to see how far the MSH diffuses, go out and tan and look at your tan lines. They can be very sharp. That shows you just how far the MSH diffuses, not far indeed. What this graph over here represents are the wavelengths that are recorded by a machine called a spectrophotometer. This machine would tell us that blue colors have lower wavelengths, whereas reddish have higher wavelengths. If we feed different colors of human skin into this machine, it tells us that our skin is basically the same mixture of 3 colors: a brownish melanin, an orangish keratin, and a maroonish hemoglobin. So that raises the question, why does our skin look so different when this machine tells us that it's really not? To answer that, we'll need to talk about the physiology of the eyes in BI 232. But for right now, I can tell you that the eyes are specialized to detect very minor changes in skin color, and these minor changes are amplified. This helps us to communicate with one another non-verbally. For instance, I might be able to tell when somebody's embarrassed due to a little bit of flushing of their cheeks. I might also be able to tell when somebody's sick. They might be a little cyanotic or jaundiced. These colors in their face are very apparent to us, and as a society, we might shun that person from our group, or if they were already a part of our group, we might try and take care of them. So while it's completely normal for humans to have very different skin colors, there are some medical conditions that can affect our skin color that may or may not need to be treated. For instance, jaundice causes a buildup of red blood cell breakdown products in our skin because the liver is failing. This is a sign that that patient needs treatment. Addison's disease can also cause a change in skin color. This is a disease of the adrenal glands that we'll discuss in more detail in the last chapter. Vitiligo and albinism are both defects in melanin production. Vitiligo can cause just localized melanin loss, whereas albinism is body-wide melanin loss. And these conditions come with significant medical considerations, especially albinism. There's going to be an increased risk of skin cancers and a number of other things that are a little bit more complicated than you might think. And that's because melanin is more than just a pigment in our skin. Next up, this is a completely different topic. So we need to take a short breather and now move on to vitamin D. This is a vitamin that is synthesized in the human skin from cholesterol. In order to do this, keratinocytes require energy, and that energy comes in the form of UV light. That energy is harnessed to convert cholesterol into an activated form of vitamin D, cholecalciferol. This can then travel to the kidneys, where it becomes further modified into the hormone calcitriol, and that hormone is required for us to absorb calcium from our diet. This is especially important when we're young and our bones are growing rapidly. Without enough calcium in the blood, the bones could not grow strong, and instead they might look like this. They bend under the weight of this person. We'll talk more about calcitriol when we get to the chapter on bones, but for right now we need to understand that vitamin D does not literally come from the sun. We use the energy from sunlight to convert cholesterol into an intermediate that becomes a hormone that we require before we can start absorbing calcium from our diet. So this page here summarizes vitamins and the skin. It turns out that melanin antagonizes vitamin D production. Too much melanin would lead to absorption of too much UV light. There would not be enough to convert cholesterol into the amount of vitamin D that we need. For this reason, people with dark skin are often told that they should take a vitamin D supplement if they move to a more northern region of the United States. It turns out that African Americans are not at a higher risk for a vitamin D deficiency living in Alaska compared to their white neighbors. Nevertheless, a vitamin D supplement is pretty cheap. On the other hand, we need enough melanin to prevent too much UV exposure. With too much UV exposure, that could increase the risk of skin cancer and decrease levels of folic acid. This would be especially bad for a pregnant female, as it'll greatly increase the risk of very serious birth defects in her child. This is just a little lesson in life. Too much of a good thing can be bad, but sometimes even carcinogens like sunlight are necessary in the right amount. We need a little bit of exposure to this carcinogen to make vitamin D, but too much could decrease folic acid. So that's the epidermis. And we covered a number of important concepts. We focused a little bit on skin cancer to illustrate the importance of anchoring junctions between keratinocytes and the lack of such in those melanocytes. We talked about the function of melanin and vitamin D production as well. That'll lead us to the dermis, a layer of connective tissue below the epidermis. The dermis is the thickest part of our skin, and it's actually two parts. The old-fashioned names are the papillary and reticular layers. These days, we know that the papillary layer is made of areolar connective tissue. It is a bit looser than the deeper layer of dense irregular connective tissue. So watch out, the reticular layer is not the same as reticular connective tissue. It is actually dense irregular connective tissue. In the papillary layer, we've got plenty of room for blood vessels, and we need extra blood vessels because this is where all of the nutrients that the epidermis receives come from. So there's a plexus of papillary blood vessels up here close to the epidermis. There's also a deeper layer of blood vessels. If any of these blood vessels becomes damaged, blood can leak into the area, but it doesn't travel very far, and that's because the connective tissue is fairly dense. So a bruise is a localized area of blood that is seeped out into our connective tissue. Blood flow to the papillary blood vessels is regulated by the brain. We can either increase or decrease blood flow close to the surface of our skin. When we want to retain heat, blood flow is restricted and instead stays deeper. When we wish to radiate heat and cool down, more blood flows close to the skin. This here is an example of a very famous vascular nevus. It's a form of birthmark. There's a lot of different types of birthmarks, but this is the one type that we can discuss based off of the physiology that we've learned. And in this type of birthmark, the nerves controlling the blood vessels did not form properly, and so blood is constantly flowing to this region of skin, making it appear reddish. It's like a constant super blush, if you will. Next up, our connective tissue has a lot of different types of protein fibers for strength and elasticity. Collagen fibers were the primary fiber type found in dense, irregular connective tissue. But there's also some scattered elastic fibers in there as well, which help to give our skin its turgor, or its ability to snap back into shape after it's been deformed. A number of things can damage the skin. One of the most common forms of skin damage comes from excessive exposure to UV light. This gentleman over here was a truck driver for most of his life and exposed one side of his face to significantly more UV light than the other, and we can see the damage that accumulated over that time. Another type of damage comes in the form of stretch marks. These can happen during the rapid growth of the skin, such as during pregnancy. When the body needs a little bit more skin, the epidermis and the dermis can usually keep up with those demands. Epithelial cells have no problem dividing, and the fibroblasts found in the dermis can make some more collagen as needed. But during rapid growth, that's where problems might arise. The epidermis has no problem. It divides and produces enough epithelial cells to prevent any open wounds from popping up. The dermis, however, has a tougher problem. These fibroblasts cannot keep up with the demand for more collagen fibers. And so the new dense irregular connective tissue that is produced is not quite as dense as the older regions of skin. And for this reason, whatever is underneath this new region of skin is more visible. If this is a highly muscular area with a lot of blood supply, this stretch mark will appear darker than the neighboring regions of skin. On the other hand, if this stretch mark is over some subcutaneous fat, this stretch mark will appear lighter than the regions of neighboring skin. To make matters worse, stress hormones block the activity of fibroblasts. And as I understand it, pregnancy can be a very stressful time on the human body. So just when you need your fibroblasts to be working harder, they're actually slowing down. And one thing that we know can inhibit the effects of stress hormones on our fibroblasts is vitamin E. So you will often find this cream in skin lotions due to its ability to reduce the damage that stress hormones can do on our fibroblasts. There's no reason to think that vitamin E could get rid of a scar. This is more of a preventative measure. Lastly, there's lots of other ways that skin could become damaged. For instance, we talked about UV light. That can just break down molecules. If it doesn't break down DNA, it can also break down collagen, which can lead to wrinkles forming in the skin. Next up, dermatitis is a symptom. It literally means inflammation of the dermis. If I hit my knee on something and my knee swelled up, I could say that I am suffering from knee-itis, in the same way that we say dermatitis. It's a very generic way of saying that there's been damage to the skin. This could be physical damage. It could be chemical damage from a toxin or other irritant. It could even be a sign of infection, such as by a yeast or bacterium. One thing that helps to protect us from bacterial or yeast infections in the skin are bacteria and yeast that live on our skin. There are a number of microorganisms who live on our skin that do not trigger an inflammatory response. They can compete with bad bacteria or other microorganisms that might induce dermatitis. Next up are cleavage lines. If we look at the dense irregular connective tissue of the dermis under the microscope, it looks like all of the collagen fibers are running in random directions. But if you were to zoom way out, there is some overall organization to the collagen fibers. And those organizational lines are called cleavage lines. If you are in surgical technology, you may want to pay attention to these, because it turns out if you cut against the grain and suture it, that will more likely lead to the formation of a scar than if you cut along the grain. So knowledge of these cleavage lines is very important for the surgeon. Those cleavage lines are not the same thing as our palm prints. The palm prints are created by extra bits of connective tissue, collagen, that helps to anchor the dermis down to the underlying tissues below, like muscle or fascia. The palms need extra collagen fibers to do this because they get more friction. Below the skin is the hypodermis, typically adipose tissue or other loose connective tissues. This is not considered part of the skin, but there's really no other chapter in which to discuss this tissue. The amount of subcutaneous fat is highly responsive to the environment, namely how many calories a person eats. This will also be responsive to genetics and epigenetic factors that we inherited from our family. Remember when we discussed adipose connective tissue, we said that adipocytes could shrink if some of those triglycerides were used for fuel, but they do not go away. Liposuction is the surgical removal of adipose tissue. Without a change in behavior, though, adipose tissue can come back pretty easily. The appearance of cellulite is created by bands of connective tissue similar to those that we saw in the palms. These can help anchor overlying skin to underlying tissues like muscle, and when they do, can compartmentalize some adipose tissue into bumpy structures that we call cellulite. A lot of people wish to get rid of their cellulite. In 2008, medical professionals spent $50 million on cellulite removing devices, and then charged customers significantly more, I'm sure. Liposuction can be one way, simply getting rid of the adipose tissue. There are a number of creams which can induce swelling. This is not always good for the body, especially chronic inflammation would not be a healthy way to reduce the appearance of cellulite. Even more dubious are some of the dermal fillers, injecting the body with plastics or other substances to try and fill up the space. I've heard some horror stories of people getting fillers done by non-medical professionals that have induced necrosis in the body. I've gone to the experts, and at this point they say while some of the treatments can provide temporary benefits, there are no good long-term treatments for cellulite at this time. So that's the dermis and the connective tissues found there. Next up are the appendages of the skin. These include hair, nails, sweat glands, and sebaceous glands. All of these structures are epithelial in nature, even though most of them originate deep down in the dermis. Hair can be found in nearly every surface of the human skin, with the exception of thick skin on the palms and soles, and a couple of other places, such as the lips and portions of the external genitalia. This man over here holds the Guinness Book of World Records for being the world's hairiest man. He does not have hair where he shouldn't, it's just that his hair is thicker than the average person's. Hair has a number of functions. One of the main functions is insulation. Being from Oregon, this reminds me of my mom telling me in winter to always wear a hat because we lose 70% of our body heat from our head. It turns out she was technically correct, but the portion where we lose our body heat is our mouth, connected to our lungs. The lungs have a huge amount of surface area and can lose a lot of heat, but you can't stop that heat loss by wearing a hat. So the thick hair that we have on our head is for insulation, but not to keep us warm. It turns out it's there to keep us cool, especially for people living in regions where there's a lot of sunlight. In Africa, for instance, many people have very thick curly hair on the top of their head, and those thick curls make for a bunch of tiny isolated air pockets, similar to the structure of styrofoam, if you will, which is a very good insulator. So the hair on our head is there to insulate us from the sun. And those of you who have an afro, your hair is much better than mine at insulating from sunlight. So each hair is created by a follicle that extends deep down into the skin. The portion of the hair itself that's below the skin surface is called the hair root, whereas the visible and unattached portion is called the shaft. Different books will draw the dividing line between root and shaft differently, so that's not a very important distinction. When we say these words, we're talking about the attached versus the unattached part of the hair itself. The hairs are produced by hair follicles. The follicles are the living cells, but eventually the keratinocytes in the follicles produce so much keratin that they die, similar to the way that was happening in the skin. Only in a hair follicle, the type of keratin that is produced is much tougher than the keratin produced in the rest of the skin. So the follicle is an infolding of the epidermis that extends deep down into the dermis, but includes a bunch of living epithelial cells. The hair follicle can be connected to nerves, which means that hairs are very sensitive to the touch. So a hair follicle does have a surrounding connective tissue layer, but then there are layers of living epithelial cells, which different books may give different names. Down at the very base of this hair follicle is a little bump called the hair papilla, and it's here that you find the melanocytes that produce the melanin that give our hair its particular color. The hair itself is composed of dead keratinocytes that came from the living hair follicle. Next up are the hair accessories. These include the erector pili muscles, which are smooth muscle that can cause the hairs to stand up on end. I don't know of any function that this serves in humans, but when it happens to my cat, it means you should leave him alone. Next are the sebaceous glands, which are associated with hair follicles, and these produce oil, which helps to lubricate the hair and reduce the growth of bacteria. The keratin that you find in a hair is similar to the keratin that you find in the rest of the skin. It's a tough fibrous protein. However, in a hair, the keratin is significantly tougher than you find elsewhere. Nevertheless, if you look at a hair, the keratin in the outer cortex is even tougher than that in the center. A hair follicle does not live forever. After 7 to 10 years, the hair follicle will stop producing a hair, and at that point, a new hair follicle begins to grow underneath the club follicle. And as this new hair grows, it ultimately pushes the old one out. This is very similar to the way that our teeth develop. We have different types of hair in different parts of the body. On the tops of our head and eyelashes and a few other places, we have the much thicker terminal hairs. In the rest of our skin, other than regions of thick skin, we have vellus hairs, which are a softer and smaller hair follicle. One form of male pattern baldness can occur when the terminal hair follicles accidentally switch over to the vellus type and produce thinner hair on the top of the head. The color of our hair is determined by the melanocytes down in the hair papilla and the type of melanin that they produce. Remember, we have two different types of melanin that melanocytes can produce, pheomelanin or eumelanin. The shape of the hair is determined by how evenly the keratinocytes divide. If one side of the hair divides more quickly than another side, the hair will be bent and curl. Just for fun, it turns out that people with blonde hair actually have more hair follicles than other people. I don't know why. The average lifespan of a hair follicle is somewhere between 3 to 7 years, depending on the person. Not everybody could grow hair down to their ankles, even if they tried, because the hair follicle would die before the hair got long enough. The keratin that we find in hair is exceptionally tough. And in fact, once upon a time, when I was trying to fix a clog in my bathroom sink, the Drano that I poured into the sink ate through the metal, leaving the hair clog intact. Hair does not grow after we die, nor do our fingernails. It turns out the skin dehydrates and recedes, which makes the hair and nails appear longer. So those are the hair and some of their accessory structures. The nails are very similar to hair follicles. Just the shape of the structure they produce is a bit different. The nails themselves are dead keratinocytes, just like hairs. They are produced from what should be called a nail follicle, but instead somebody named it the nail matrix. But this is the same thing as a hair follicle. It is the living cells deep down in the dermis that produce the non-living nail. The portion of the nail that we can't see is called the nail root, whereas the rest of the nail would be called the nail plate. It turns out that our nails do not grow at the same speed. Our fingernails grow faster than our toenails, and the nail of our pinky grows slower than the nail of our index finger. This keeps all of our nails relatively the same length. Lastly are the glands, which come in two major flavors. The sebaceous glands we've already discussed. These can be associated with hair follicles and produce an oily secretion, whereas the sweat glands produce a more watery secretion. Both of these are exocrine glands, meaning they dump their substances to the surface of the human body. So the sebaceous glands produce oil. They release that in a holocrine fashion, if you remember that word from the previous chapter, meaning that when the cells produce the oil, they actually die. So this will require cell division down deep in this gland to replace the dead cells. The oil that they produce is called sebum, which contains a number of lipids, which makes it oily rather than watery. These lipids help to lubricate the skin and hair, and also controls the growth of bacteria. The sweat glands are more accurately called the merocrine or the eccrine sweat glands. These produce sensible perspiration, meaning I can feel the fluid as it's released onto the surface of my skin. We do this to help radiate more heat off the human body. As that fluid evaporates, it takes body heat along with it. The sweat produced by these glands is mostly water, plus a bunch of electrolytes and some waste products. So the major function of sweat is to cool the skin. It can also help to flush away microorganisms. Our feet have 500,000 sweat glands each, which all together can make about a pint of sweat per day. All the more reason for your children to change their socks. There are a few specialized sweat glands that we must also discuss. The apocrine sweat glands are very similar to the merocrine sweat glands in appearance, but their function is different. The sweat that they produce contains a larger amount of lipids, which, when they break down, give us body odor. The reason that we have these types of glands is for nonverbal communication. These glands only become active after puberty, and the body odor that they produce can act as a pheromone, alerting other humans that that person is now ready for reproduction. It may seem strange that body odor and sex would be linked to one another, but you just have to keep in mind the context. Body odor can be very annoying when it's somebody that you don't like standing a little bit too close to you. But when you are having sex with your partner, you really do not mind their body odor, and in fact your brain would interpret that as a very good thing. Do not expect to recognize the difference between these under the microscope. We can identify the apocrine sweat glands based off of their location. We find them only in the armpits and around the genitalia. There are two other modified sweat glands that we will talk about later. The mammary glands are actually a modified sweat gland, and earwax is produced by another modified sweat gland. But those will be topics for different classes. So that wraps up the accessory structures of the skin. So that leads us to the last topic of the lecture, which is how damage to the skin gets repaired. It turns out that the processes here are very similar to damage to any organ. So pay close attention to this because it'll be relevant throughout the rest of the year. First off, if any blood vessels are damaged, blood loss must be limited, and a scab or a blood clot must form. We will learn about this in much greater detail in BI 232. Next up, very rapidly, the inflammatory phase occurs, and we covered this in the previous chapter. If you do not remember the 4 hallmarks of inflammation and why they are useful, you need to go back to the previous chapter and refresh your memory. Inflammation happens very quickly. The next 2 phases, the proliferative and remodeling phases, occur weeks to months later. So it's a completely different time scale than hemostasis and inflammation, which happened within minutes of the injury. So we are going to focus on the proliferative and remodeling phases, keeping in mind first that this time frame is completely different from inflammation. So here is a simplified cross-section through the skin. When I get damage, we must first form a blood clot and then go through the inflammatory response. I'm not showing inflammation here, but inflammation will help bring in white blood cells, which can remove some of the debris, cleaning this up, setting the stage for regeneration. It turns out I'm going to need even more blood than the inflammatory response provides. So one of the first things that happens in the regenerative phase is to grow new blood vessels in this area. The growth of new blood vessels is called angiogenesis, and this will cause the wound to appear reddish. This is a completely different redness than we see with inflammation. That was caused very quickly by vasodilation of existing blood vessels. Here, in the regenerative phase, the redness is caused by growth of new blood vessels. This extra amount of blood will provide us with the nutrients that we need to grow new tissue. The epithelial tissue can grow pretty well on its own. So the epithelium will fill in the wound quickly. New mesenchymal stem cells will have to migrate into the area and differentiate into fibroblasts to produce new connective tissue. If this was a really large gap, I would fill it in as quickly as possible with scar tissue. Scar tissue is very similar to dense regular connective tissue. It's composed primarily of collagen fibers that all run parallel to one another. But unlike dense regular connective tissue, in scar tissue, the collagen fibers are cross-linked. So the new connective tissue that I've produced is not the same as what was there previously. This is not dense irregular connective tissue, and this scar will appear different from the rest of the skin. It might be a raised bump, but it still appears red because of all of these extra blood vessels. So the last phase is the remodeling phase. I've filled in the gap. Now I need to replace it with what I actually want. So I'm going to get rid of those extra blood vessels. They are no longer needed, so they will go through programmed cell death, which is called apoptosis. I don't want cells just dying, releasing lysosomes. So instead they commit cell suicide and are removed. Next up, more mesenchymal stem cells can migrate into this area and differentiate into fibroblasts, which may be able to replace that scar tissue with dense irregular connective tissue. If that's the case, I would not have a permanent scar. But in really large injuries, we cannot replace all of the scar tissue with the tissue that we actually want, and so some cross-linked collagen fibers might be left over. At that point, the scar would appear different from the rest of the skin. It would appear more whitish. It's white because the connective tissue is different, and I no longer have all of those extra blood vessels. That would be happening months and months after the original injury. So a new scar often appears reddish because of the extra blood vessels that are produced, whereas older scars might appear more whitish because the blood vessels have been removed and the collagen found in scar tissue is arranged different from the collagen found in dense irregular connective tissue. The other type of scar that we already discussed earlier were the striae or stretch marks, and those are caused by a change in the density of the connective tissue, not in the amount of cross-linking between collagen fibers. So here are pictures of a scar at two different time points. This reddish scar is going through the regeneration phase. It appears reddish because of the increased number of blood vessels that have grown into the region of damaged tissue. But after months, or maybe a year, we are left with a more whitish appearing scar. The scar tissue looks different from the rest of the skin. We have removed those blood vessels after they were no longer needed by the process of apoptosis, or programmed cell death. Lastly, you may need to estimate the amount of skin that has been damaged. For instance, if somebody has suffered a burn. A first-degree burn involves damage only to the epidermis, and a sunburn is a common type of first-degree burn. The damage of a first-degree burn can heal very easily because there are plenty of epithelial cells. A second-degree burn includes damage all the way down into the dermis. This is obviously worse than a first-degree burn, but still can heal itself relatively well because there are plenty of epithelial cells down in the hair follicles, which, if needed, can divide to produce new stratified squamous epithelium covering the surface of the skin. A third-degree burn completely damages even those epithelial cells way down deep in the dermis. This type of burn could only regenerate from the edges, and therefore a large one especially would require a skin graft to help speed up this process. Skin from another part of the body can be cut into tiny little pieces and seeded over the injured area, similar to the way that a lawn might be seeded, to help speed up the regeneration process. A significant amount of burn is very dangerous to a person because of the major function of the skin, which is to provide a barrier, a water-resistant barrier. Without stratified squamous epithelium, water evaporates from our body very quickly. And so one of the biggest concerns for a burn victim is water loss, and you will have to rehydrate them with an IV. Our second issue of concern for somebody who's lost a large portion of their skin is infection. That shows us the second major function of the skin, to provide a barrier between the outside world and our inner tissues. We can worry about preventing infection after we've gotten dehydration under control. So that wraps up injury to the skin. We had those two very quick responses, forming a blood clot and the inflammatory response, and then two much slower processes, proliferation and remodeling.